pull it up. I do have it. So let me share screen. Sorry, iPhone, you don't see. I was in a Zoom meeting on my phone, so I was able to just listen. No, this was my meeting yesterday. So I'm like, well, I can't see what you guys showing. All right, so we're on the practicum, the mock practicum on my Canvas. Raymond, would you like to point us where we're going?
Yes. I know the microscope parts are kind of familiar, and I kind of know them, so I don't really have questions on that. But I don't know. I don't speak for myself. I don't want to speak for myself. I don't know if other people in the class might have this. So if you guys want to ask a question on this, like.
For the student on the iPhone, we're looking at pictures that have a microscope, and on the microscope we have different letter taped to the different parts of the microscope. And so the question is to name the structures and provide A functions. Please be very specific, you guys. On the practicum, you gotta give me detail. Detail show me that you understand stuff. Okay, so you have to be detailed.
So for H, wouldn't it be like the y-axis, and you could just say that it moves it can move the sage vertically or horizontally?
You know? Exactly. If you say that, that's the full credit.
Okay.
For the E and F, which is pointing at the coarse adjustment knob and the fine adjustment knob, you cannot just say it focused the specimen.
Yeah.
And I say, well, so does the objective lens.
Yeah. So if I say this for the course, you usually use the course for when it's when you're using the 4x objective lens, and you could say for the fine, you could use it more when you want to, like, for a more sharper, finer focus.
Exactly.
Okay.
Give me that, and if you give me that detail, then I know you're able to separate an objective lens from a fine adjustment knob and from a coarse adjustment knob. You know what I mean? Yes. I'm getting into your head. Yeah. To say, okay, do you know the difference? Okay, you tell me that, then I say, Raymond, you got it.
Okay.
So then I give you full point.
Yeah, that's what me and Savannah and Maribel and Aaron have been trying to, like, give as much detail as possible. And then I think tonight we're gonna do like a timer. We're gonna each person gets a slide, and we're gonna do 4 minutes and without looking at notes.
Exactly. And you can grade each other.
Yeah.
But this is detail enough. No, you left this out. So that's why I like the group study a lot. Because you can assess each other before I get to assess you.
Yes.
All right. So the next slide will be, so the question is, what are the 2 ways you can adjust on the microscope to get a clear image? Which filter would you use to view like a wet mount, a live specimen? And then asking about what do you need to do when you get ready to put away your microscope.
So for question part A, can we just list them, or do you have to also provide a description on it? Because it's just provide.
So for a question like this, think about, put yourself in my shoe. You are now tasked to teach someone how to use a microscope.
Correct.
Which you guys already do in that, right? So how would you teach that person? You know?
Yes.
So make it clear in writing. How would you teach the person the 2 way to help them get a clear image of their specimen?
Okay, I get what you mean.
Yep. Okay. Moving forward, we have quadrant streak. Is it okay if I go slide by slide, or Raymond, you want to jump to a certain one?
Oh no, it's perfect going slide by slide. I don't mind.
Okay. So you might hear this again on Monday if someone asks, okay, but we might not have time. So so the next slide for my student on the iPhone is we have plates, a quadrant streak plate, okay, and it's labeled ABC. And then so it asks, what is the purpose of performing quadrant streak? And then the next one is looking at this plate, which one is the best, which one is okay, and which one is the worst. And then once you decide, then how can you help the student improve their technique? Okay, on you decide what plate needs improvement.
So for this one, would you consider the best being letter B, and then the second best would be A, and the last one would be C?
To be honest, if I'm looking at this, yes, you're right. The best one would be B, but then you have to tell me why you picked B.
Okay, so why? Because the reason why is because they got single colonies, and then for A is they at least they backtrack to fill in all the quadrants. But C, I think they heated up their loop too much and they didn't backtrack, so there was no growth at all in the other two quadrants.
If I have to rank them, I would say B, C, and then A. A is actually the worst.
Okay.
Because they probably didn't flame their loop, which I saw some of your classmates have this type of pattern. They didn't flame the loop between streak. And then even worse, they dipped back into quadrant 1 when they were doing, or they dipped back in quadrant 2 when they were streaking in quadrant 4.
Okay.
And C is not ideal. I see that because you don't get any single colony. But at least, well, I don't know. C and B is about on the same the same level.
Okay.
Because you're not getting single colony.
You have mixed culture, right?
Right. So you can say that. You're like, I can't decide, Professor. C and B are about almost the same because you're not getting single colony.
Okay. Thank you.
You know? Mm-hmm. Yeah. As long as you explain to me, and I'm like, well, yeah, that makes sense.
Mm-hmm.
You know?
So in a way, there is no right or perfect answer. It's just you want to test our knowledge to see if we could explain it and basically give the detail.
Exactly.
Okay,
that is my ultimate goal, is to assess your understanding of the material we present to you.
Okay.
Okay, number 4 is on a Gram-Stain slide. And then one of the question asked, is this a pure culture and how can you tell?
It would not be a pure culture because there's different morphology. There's like a bacilli and cocci.
Exactly.
And there's also...
You did that and you get full point.
Okay. And then wouldn't the mordant for this one, for the Gram-Stain, be iodine, and the reason is to adhere the primary stain to the slide?
Exactly.
Okay.
Okay. And so that's question C, is what is the mordant and why do you use the mordant? Okay. And then B because this is not a pure culture, because they have different morphology, like Raymond said. So then I follow that with pointing to different bacteria, and then I ask you for the shape and arrangement. I could do that on the actual microscope as well. I have a pointer, so I could point at something and I'll ask for a morphology.
Okay.
So that's why be very careful when you come up onto the microscope, because if you accidentally bump into it, it will point at something else.
Okay.
Yeah. If that happened during the practicum, do raise your hand and say, Professor, I don't think it's pointing where you want it to be pointing at.
Okay.
And then I'll take a look and I'm like, you're right, I need to adjust it or something.
Okay, for this one, this is the slide that me and my group were confused on. It was the tube, which we don't remember what test this one was.
Okay, so one tube is yellow and it has the layer of oil on top. The other tube is purple and it has the layer of oil on top. So this is the decarboxylase test.
Decarboxylase test, okay.
So yours is not that purple. Remember, you're like, I can't even see.
Oh yes, I remember we said that it wasn't that purple. We're like, oh, did we do it right?
Right.
Yeah.
So that's the test.
Okay.
Okay, so A would be a negative result, no decarboxylase, because there is no change in the color or the pH.
Okay. So that's all we can say, like, oh, test A would be negative because there's no change in pH or color.
Correct.
Okay.
So don't stop at saying no change in color. I mean, I see that.
That's what
what does it mean?
We're trying to limit ourselves. Like, if it gives like a why, because like for the Durham tubes, like why is there oxygen, or like why do we use the Durham tubes? Like we say the reason why we use the Durham tube is to see if the sugar fermentation produce a sort of gas.
There you go.
That's what we're trying. We're trying to give as much detail. We just want to say, oh, it changed color, and that's it. We're trying to give why it changed the color.
Correct. Okay. So you notice I just said interpret result. I didn't say explain yourself.
Okay.
Right?
I know, but
interpret
I
result
just think it's
means
just like
explain yourself.
yeah,
Explain why you say that is the result.
and I just feel like that's just part of me, like myself, like my self conscious. I'm like, I need to give so much details because I don't want to miss out on these points. You get me?
I see that with you, Raymond, and I totally appreciate that.
Yeah.
When I'm grading your answer, I'm like, yes. Like he explained himself, you know? So
Yeah.
I totally appreciate that.
Okay, thank you.
I like that.
And then for the slide, me and Savannah were kind of having a little debate on this, but wouldn't this one be the acid fast one?
Why would you say that?
Because this is the pink one, and then we use methane blue. And I remember we never used methane blue for any side. We just used it on our first one for the simple saying because we could choose any color we wanted.
We use methylene blue for an acid fast.
Yeah, that's what I said. We used it for the acid fast. Yeah. But the primary saying for this one wouldn't be the methane blue. It would be the carbofuchsia, because that's when you heat it up.
Right. And the bacteria kind of stick together because
with
on the surface
with the egg whites. With
malic acid.
Yes.
Yep. Okay. And then it asks for so the second part of this slide has a picture of bacteria, and then it asks what type of stain is on the slide. So Raymond say this is an acid fast stain. How do he know? 2 reasons. One, the background is blue, and we typically use methylene blue as a counter stain. And then the second reason is the bacteria is clumpy and is stained pink. And so for acid fast, we use carbofuchsians as the primary stain, which stain bacteria pink or red. Okay. The next slide, I have a paramecium, and it could be anything. I could switch it up. I could use a pinworm. Okay. Mm-hmm. So it doesn't matter what I put on. But if you look in the microscope and you see the organism being lined up to the ocular meter, I'm going to ask you what is the length or the width of the organism. So very similar to your soil unknown project, we've been asking you to measure the length and width of your bacteria. So I'm assessing the same skill. Okay, and then the other one is asking, so be mindful, because this one is not on the 100x objective lens with oil. With the soil unknown, we are measuring your bacteria on 100x plus oil. So every increment on the ocular meter is equal to 1 micrometer.
And for this one, it's 2.5, right? We're measuring
it on the 40x.
So it's 2.5, right?
Correct. So that you need to know.
Okay.
It will not be provided.
Okay. And for an example, would we be able to have a calculator present with us to do calculation?
Yes.
Okay.
Thank you for asking.
That's what I was kind of confused on. We're like, oh, wait, she could give us a calculator because there's some things that we probably need a calculator for.
Remind me on Monday to say that.
Okay, because
you guys are welcome to bring a calculator.
Okay.
Just leave it in your pocket as you rotate.
Okay, thank you so much.
But you need to show me the work, okay?
Yeah, like write out the equation, but just like use the calculator for it. Okay, makes sense. Thank you.
And then the other question asking is, what is the total magnification?
Oh, it'll just be 400, right?
Right. Show me the math, because it's 40 10 from the ocular times 40 objective lens is 400.
Okay.
Okay. Jump in anytime, everybody. Raymond and I just go through the slide. Jump in anytime. The next one is a plate. And on the plate, you see bubbles on the culture. Okay, so what test is that?
The catalase test?
That would be the catalase test. Okay, and so...
But I was we were kind of confused on it because you gave 2 of the same catalyst tests on the practicum, on the mock practicum. Like this one looks like, I thought we were kind of saying this was the oxidative test because it kind of looks blue. Mm-hmm. But if you go to like more slides, I don't remember what slide it was, but there's another one that looks like the catalase test as well.
Oh, I don't know what I did, but so this was my student project. Actually, is the soil unknown project. Okay. You guys did this.
Yes.
Right. And so on you guys did catalase on one side, oxidase on the other side.
Mm-hmm.
Well, this student decided to do catalase and oxidase on one side.
Okay.
So if you see bubble, then it's a catalase test. If you see purple, then it's an oxidase test.
Okay.
I will not do that to you on the practicum. But then again, I'm not the one setting up the practicum. So we'll see what's being set out for you.
Okay.
And then the other one we haven't looked at, it was a gelatinate test.
Yeah, and this
Right?
is the one.
We have to wait 7 days.
Yes, and then this is the one that you said that it's like it increases its virulence. I'm sorry if I'm butchering the name, but the reason why it's like it increases because they eats our connective tissue and it could like spread quicker, right?
Yes, exactly. So for the student on the iPhone, there is 2 tubes, and then the tube kind of leaning 45-degree angle. One tube looks solid, and the other tube look liquidy. Okay, so the name of the enzyme is gelatinase, which is a component of our connective tissue. So if a bacteria able to make the enzyme gelatinase, it's going to break down gelatin into single amino acid. That's why it look liquidy. Okay. And so the other question is, how does this bacteria use this enzyme to increase its virulence? Virulence is the ability to cause disease. Okay, so the way, like Raymond said, it increased their virulence because they're able to kind of eat through your skin. Your skin is a good barrier because you have connective tissue that prevent bacteria from penetrating. If this bacteria able to eat through your connective tissue, then obviously it penetrates through your skin, and now it go into your bloodstream because our skin is very vascular. Okay, so once it get through, it has access to our circulatory system, and now it could become a systemic infection. That's why it's kind of scary. Okay. The next slide is containing thioglycolate tube with tube ABC with different growth pattern, and I'm pretty much asking you to please name the oxygen requirement for each of the organism. Okay, and so Raymond, you would like to go through, or anybody would like to tell us the requirement for organism A, B, and C?
For A, that would be strictly anaerobic, correct?
And so tell me why. Well, tell the person on the phone why.
Yes. Okay. So if you're looking at this tube, you can clearly see that the top portion, there's no growth. It's all below. So therefore, it does not need oxygen to grow.
It get killed by oxygens. So it's staying away.
Yes, because it's scared of it.
Yep.
Do you want us to tell you why it is that, or can we just put strictly anaerobic, aerobic, and that would be okay?
So I'm glad you asked, because look at the question itself, right?
Okay.
This question just say name the oxygen requirement for each of this organism. And so you just frankly just say this is a strict anaerobic. However, if you look at my question, it say interpret results. Then you need to give me an explanation of why you say organism A is a strict anaerobic.
Okay, I got that.
Yep. So how the question kind of being asked. And it's okay. On the exam, you can ask me that. Like, do you need me to explain? And then I would say yes, or I would say no, I just want you to tell me the type of oxygen requirement.
Okay. Yeah, that's fair. Thank you.
Okay, so for the phone, to B, what type of oxygen requirement would that be? Not the person on the phone, the people here who can see it.
Wouldn't that be strict aero?
And please tell the student why you say that.
Because it's growing on the top, so that means it requires the oxygen to grow.
Grow on the top. only,
Yes.
right?
There's only growth on the top, so that means that the oxygen was present for the bacteria to grow.
Exactly. How about C? How about Gabe? Could you please tell the student on the phone? Student can't see, so.
Vacuolet vanderope, right? Because there's growth on the top and going down as it descends.
As it
Correct.
descends, what happened to the amount of growth?
Oh, it did. It became less dense.
Exactly. Okay. So we have to clarify that, because if I have a tube where the bacteria grow everywhere, there is no different in the amount of bacteria grow, then what oxygen requirement would that be?
Can you
If
repeat
I
that?
hand you a tube that like just uniform grow throughout.
Wouldn't that be the tolerant one?
Yeah, it'll be aero tolerant.
Aero tolerant. There you go.
So I have to know that you know the difference between aero tolerant and facultative anaerobe. The growth pattern is slightly different. You guys see why I asked for detail?
Yes, you want to be able to see our understanding on the
Right,
material.
right. That you're able to differentiate one from the other. All right. The next one is still on oxygen requirement, but this time we have plates. And just like you guys did in class, the plate have 4 different quadrants. And so now I'm asking the same thing. Determine the oxygen requirement for these organisms. And then on the plate is ABC, ABC. So you have to look at the growth density across the plate for organism A, and then that will help you determine the oxygen requirement. Okay, so just for an example, I look at the plate, and organism A seem to have a slightly higher growth on plate A, then plate B, then plate C. So like Gabe answer to the last two, there is a gradient in growth. Okay, so heavier in air, a little bit lesser in the candle jar, and a little lesser in the anaerobic jar. Okay, so then if that is the description, what is the oxygen requirement for organism A?
Facultative?
Yep, facultative anaerobe. Okay, so it's almost the same pattern of interpretation, but we're still looking at density of growth, but now we're looking at the plate and what condition the plate has been incubated in. So if you look at organism C, right, there is no growth in the air plate. There is growth in the plate that was in the candle jar, and then there's no growth in the plate that we had incubated in the gas pack jar. So what oxygen requirement would C be?
Wouldn't that be microanerophilic?
Right, because the candle jar is supposed to give us the microaerophilic environment. Okay, and so you only see grow in that plate, then that tells you the bacteria is a microaerophilic type of bacteria. And then the follow-up question is, okay, well, what makes the candle jar a microaerophilic type of environment?
The oxygen decreases and the CO2 increases.
How?
Because obviously we know that the candle requires oxygen to to basically keep burning, but since eventually the candles get turned off and it's going to release the CO2.
Right, we're burning something, right? So when we burn something, we release CO2, and to have fire, you need oxygen. And so when the fire go away, that means our oxygen level also going away. The next slide is a set of tube. We haven't done this one, you guys. This is the nitrate reduction tube. Okay, so for this one, please look at it carefully. So if you look at tube A and B, you see those powder. Okay, those are zinc powder. Okay, so those are zinc powder. So on the practicum, make sure you look. Is there any sign of zinc being added? If it's not on the side of the tube, look underneath the tube. Is there any signs of zinc being added? Zinc is going to react with nitrate. And when zinc met up with nitrate, it's going to change the broth color to a red color. So with that said, and we're looking at, does the bacteria use nitrate? So if you look at tube A, you see zinc and you see the broth is red. That alone tell you that the organism did not use nitrate, because the nitrate react with zinc, give you a red color. This tube had zinc and red. That means the organism did not use nitrate. They do not have a nitrate reductase enzyme. Versus tube B. Okay, tube B, we added zinc, but the tube did not turn red. Okay, so that's telling you, I don't know what happened to nitrate, but they're not there anymore because the zinc couldn't find any nitrate to bind to cause a color change. Okay, so you say positive for nitrate reduction. The bacteria do have the enzyme nitrate reductase. However, I'm going to ask you further, okay, because I'm going to tell you the information. If you look at the Durham tube, there is a big old bubble. Okay, so I'm telling you now, we're going to go over it in Monday lab. Okay, where's the nitrate go? The nitrate had changed into a gas because I see a big old bubble in the Durham tube. What are the gases? The gases can be nitric oxide, nitrous, or nitrogen gas. I don't know what it is. I have to do more testing, but those are the possibility. Okay, so the questions say interpret result. So you have to give me all of those information to get full points. Okay. And then tube C and D, you don't see any powder. If you look underneath, there's no powder either. Means zinc was not added. But they add two other indicators. All of these have three indicators. Okay, tube A and tube B. Okay, all of these tubes, they have okay. Let me backtrack. Tube A, tube B, and tube C had indicator added. Tube D did not. Okay. So on Monday, when I asked you to pull your tube out, the first thing I'm going to ask you is, look, is there any bacterial growth? Is it turbid? Okay. If there are turbid, please look at the Durham tube. Do you have any bubbles in the Durham tube? Okay. If your tube has bubble in there, you don't have to add any indicator whatsoever. You say, that's a positive result for nitrate reductions because I saw a bubble in the Durham tube as soon as I pull the tube out of the incubator. That bubble is nitrogen gas. So nitrate, NO3 minus 1, had changed into nitrogen gas, which is N2 gas. Done. That's a positive test, and not add any reagent to it. If you do not see any bubbles, okay, go ahead and add reagent for me. There's two reagents that I would like you to add. Sulfonilic acid, followed by, I think alpha, or is it dimethyl alpha naphthoamine? You need to know that name. Okay?
Can you repeat that name, Professor?
You're gonna see it on Monday, but the name is dimethyl alpha naphthoamine. That's one reagent. The other one is sulfonilic acid. So if your tube come out and you see growth and you need to add reagent, I'm going to ask you to add 3 drops of each into each of the two. One after the other, doesn't matter what order. Okay, then you wait a minute or so. Okay, if your broth turn red after you add that 2 reagent, okay, then that is a positive result for nitrate reduction. What is it reducing to? It's reduced nitrate into nitrite. So NO3 minus 1 become NO2 minus 1. So it did reduce. The organism did make the enzyme nitrate reductase. So that's a positive result. Okay, the other 2 tubes, there is no color change. Okay, and there's no bubble. So then I say, okay, let's go ahead and add zinc to them. Okay, this on the PowerPoint slide is the result, the color change after you add zinc. Okay, so you add zinc and tube A, give it a minute, tube A changed to a red color. Okay, so that means tube A there is a negative result because zinc react with nitrate and change to a color red. Okay, so tube A do not have nitrate reductase. However, tube B, when I add zinc, again remain colorless. That means there is no nitrate in the broth because otherwise zinc would react with nitrate and give me a red color. It did not. So the nitrate is not there anymore. Okay, so I waited a little bit, a day actually, or 2. I finally get a bubble in the derm tube. This is a slow reaction. Okay, so this bubble wasn't there when you pull the tube out. But after I add the 2 reagent and zinc and I left it for a day, I have now a big, huge bubble in the derm tube. Okay, the bubble is gases, nitrogen gas, a nitric oxide gas, or a nitrous gas. This is a slow reaction. Okay, so I have to wait and then it show up. So on the practicum, these two have been sitting around. And so I use them to ask you the question. So nitrate reduction is a little bit complex. So you have to be able to explain it to me so that I say, okay, you got it. So you see, just saying the color change doesn't mean anything. Okay, because this color change and that color change have different meaning. This color change and tube A and tube C both turn red, but they have different result. Tube B and tube D, there's bubble and no color change. However, one have reagent added and one did not. Have different meaning. So telling me there's a color change and telling me there's no color change and stop there doesn't tell me your really understanding of what's going on inside the tube with the bacteria. So for nitrate reduction, you gotta carry it forward with more detail in your answer to tell me you got it. So that's my lengthy interpretation of the result, but you guys have 4 minutes, so you have to find a better way of answering it. Okay, in 4 minutes. So the best way is practicing. Go with each other. Hey, is this answer enough? Does this give me enough detail, in the 4 minute or 5 minute that I gave you? I know other instructor gave 4 minute and that's it. There is no second round, and then there is no like another 15-20 minute after. I know that for the fact because the instructor asked me. She's like, I only give my student 4 minutes. Well, how many minutes do you give your student? Okay, so I'm like, wow, 4 minutes only? Ugh. But that's the instructor way of teaching. So I like I say, I'm walking the middle line. Okay, so the next slide is MR test, which you guys haven't done yet. It's going to be Monday. Okay, on Wednesday, which is inoculating. Okay, the MR test is to look to see if the bacteria are able to ferment the carbohydrate and produce a mixed product that are acid. Okay, so on Monday, I'm going to ask you to look for growth. Do you have growth in both of them? You do? Okay, we're going to add 2 or 3, 3 or 4 drops of an indicator called methyl red. Okay, so the same. The name of the test is methyl red. The indicator is methyl red. So when you add one of the two, the methyl red is going to disappear. It might not be that yellow. I think this making it very obvious. It might not be that yellow. It might be the same color as the broth color. But the point is the methyl red didn't stay. It disappeared. And if you add to the other tube, the methyl red stay red. When that happened, the tube that stay red is has a positive result. There is a change in pH. This is fermentation. So there's a change in pH. The methyl red stay red because it's an acidic environment. On this side, the methyl red did not stay red because it's a basic environment. So no fermentation happened there. Okay? The product is a mixed batch of acid. And that's why there's a change in pH. And that's why the methyl red stay red. And the other tube is the OMPG. I don't have to describe because the person on the phone left. Okay, the other tube is OMPG test. So the reason that the best way, if you come up and I say, what test is this? And you see a disc, that's definitely going to tell you that's an OMPG test. Because of all the tests we do, that's the only one that we add a disc to. Okay? So it's an OMPG test. And then I say, what is the purpose of this test? Is to see if the bacteria are able to use OMPG, which is a different form of lactose. And they say which test is positive. The test that turned yellow is positive. Again, this is a pH change. Okay, because the indicator is an orthonitrophenol. Is a pH change. Okay, a color change because of a pH change. Acidic result. I think it's pyruvic that's being formed. Double check me on that. Okay, so the tube that changed yellow is the positive.
I was confused because we were trying to look up the answer to see why we what does the disc contain and why do we use a disc?
So to clarify, the disc contains the indicator. What is the name of the indicator?
The
O-nitrophenol.
O-nitrophenol, okay.
Right. The broth contains OMPG.
Okay.
So if you don't add the disc, both of your tube gonna look like this, clear, like growth, and that's it.
So the reason why we add the disc is because it's an indicator?
Yeah.
Okay.
It contains the indicator. I think it says in the package. By the way, everything I talk to you here, you can find it in writing in your package. In the introduction, each of them has a paragraph, and it did not tell you the reactions. I provide you with the reactions on the board and also on the PowerPoint slide. I think I provide that. But in the lab package, it'll talk about it. Okay. maybe the reaction is in like wording, and I gave you the reaction form. Also, in the lab, those photo atlas has all of these and how to interpret result. And if you're at home, then visit Microbugs. So micro and then BUGZ, that will do the same thing like we're doing now. So those are all the resources. If I'm not around, please use those to help you. Okay, so on the practicum again, sometimes we don't give you both. Sometimes, when we inoculate, we have an A and a B so that you guys can compare. Well, sometimes we're like, okay, let's see if you can identify it by just giving you one of the two. Okay, so first would be, well, I didn't ask what test this is, but in order to answer the question, you have to know what test this is. We haven't done this yet. Okay, we'll do this on Monday. This is the tryptophan hydrolysis test. You are going to add an indicator. The indicator is called Kovac reagent, the scientist name. You add it in, and on the screen it shows a greenish yellow ring on top. That is a negative result. Okay. The tube you're going to have will have a red ring on top. That red ring will be a positive result. The enzyme is tryptophanase. The product is in the name because the name of the test is tryptophan hydrolysis or indole production. So you know one of the product. The product is indole. Okay. The other product is pyruvic acid. So sometime if you know the name of the test, the answer is in there. Stop me when you have questions, okay? Because I'm gonna scrolling along because the time is 11:41. Okay, so this question you have two slant. Okay, one slant have a greenish color to it. We done this, right? You add ferric chloride to it, and it turned green. That tube is positive. Okay, for deaminase, the enzyme deaminase. Okay, why does it turn green? Because the phenylpyruvic acid going to react with the iron of the indicator and give you the green color. So that's the question, what caused the color change? And then what is the function of the enzyme? It's deaminase, so removing the amino group from the phenylalanine amino acid. What is the other product besides phenylpyruvic? Ammonium. Because once you remove ammonia, it's going to pick up a hydrogen and it become ammonium. Just that's the reaction that it does.
So for even those questions, Professor, for like E, like you just went over, do we need to just put ammonium or you want the full? Okay, perfect.
Ammonium. Yep. So students lose point because of lack of detail. So please provide detail, provide correct detail. Okay, sometimes I get detail and I'm like, yeah, that's not right. This is determined volume. Okay, so same as the quiz. Do you guys know how to do volume? You either count up or you subtract down. This is a 5 ml pipette. and I'm at the 3. So if I go all the way to 0, the whole thing will be 5, right? So I can take 5 and subtract 2 plus subtract 2.5 or 2.6. That will give me the volume. So that's one way of doing it. Or go to the tip of the pipette and count up. Whichever way you want to do. And then make sure you're able to do unit conversions. You have a calculator, but please do show your calculations. Or move your decimal. I don't care how you do it. You're like, Professor, I know I'm going to move the decimal. Something to say, I see what you're doing. Okay, we inoculate this, but we didn't interpret result. Okay, so one of the questions I asked related to chapter 6 or the first lab is this a broth, a slant, or a deep? And then you said this is a slant. Okay. And then name this test. This was the Simon's citrate.
Simon's, yeah.
Go ahead, Raymond.
It was a Simon's test, right?
Yeah, Simon's citrate utilization test.
And
name the indicator.
I remember something like, it starts with the B.
Bromthiamyl blue. There's 2 of them start with a B.
Yeah.
The decarboxylate is bromcresol purple. This one is bromthiamyl blue. And they're both pH indicator. Okay, so instead of asking you which tube is negative and which tube is positive, I'm trying to be a little bit more creative. I say explain the color in B. So you would say, well, it's a positive result. Okay, there's a change in pH. Okay, this is also citrate has been broken down into an acid and ATP. This is the Krebs cycle. Okay, there's a change in pH, and therefore the cobalt blue color. The enzyme the bacteria had the enzyme citratase. Make a checklist. Did I hit all of these in 4 minutes? If I did, then I'm good. Okay, so I could ask like this. That's why I asked you to make sure you know the chemical reactions. So I can leave it blank. Okay, and I say fill in that blank.
And that'll be ammonium, right?
Yeah, ammonium, right? We talk about this, right? And then I could ask, what is the enzyme? Urease. Okay. Okay. This is old. This is way back when we still able to purchase a disc. The same with ONPG. Well, similar to ONPG. This disc has an indicator on it. The indicator is phenol red. Okay, and this disc also have urea on it. But over the year, Bio-Rad or wherever we buy this stuff from, they say, well, we're not making the disc anymore. They discontinue. And so Gabe ended up making a broth with urea in it. He dissolved urea in the broth, and then he added the indicator phenol red in it. So for you guys, there was no this. Okay, there was just a red, or not a red, a orangey color because of a pH difference. And orangey color broth, very small volume, and then we ask you to inoculate your organism. Okay, and then one of your tube gonna stay orangey, and then the other one give you that magenta looking. Okay. So when you do your unknown, okay, remember the result D, the little D or W, so that you can look at how how magenta is it? Is it a deep magenta? If it's a deep magenta, then it's 100%. This happened. If it's a light magenta, like kind of like pinkish, then it's a W result or a D result. You're going to see that when you do your soil unknown or your staff unknown test. Okay, so the shade of the color means something. Okay, so you have to do that as well, but not on the practicum one. Okay, we're gonna give you a dark color. Like, is it there or is it not there? Okay, so interpret results. So A would be a negative result because there's no color change. There is no change in pH. The bacteria do not have the enzyme urease. Versus B, there is a color change, there is a change in pH. The bacteria have the enzyme urease. You could even go further and say urea didn't break down to ammonium and carbon dioxide, just to really show me you know your stuff. But you don't get extra credit for it. Okay. This one is, name this test. Okay. Raymond, can you name this test?
It's the sugar fermentation test.
Exactly. Okay, it's a sugar fermentation test. What is the indicator?
Is this one the methanol red?
No.
Oh, the
this
phenol
one has
red.
A term too. One more time.
Is this a phenol red one?
No, phenol red.
Phenol red.
Okay. What is the purpose of the derm tube? You already said this.
To see if the bacteria produce any sort of gas.
Yep. Okay, name 2 possible products.
Either an acid or an oxygen, or a gas. I mean.
Yeah. Or so 2 of the 3.
Yeah.
Okay, it's in acid and alcohol or gas. Now, we didn't see this one, but I talk about it. Okay, you see there's some yellow, and then orangey, and then red. Okay, so if you see this is a D result for fermentation, the letter D. Okay, it's not 100% like 2B. Okay, it's kind of a little bit orangey. So the bacteria can ferment the sugar, but not 100%. 11 to 89%. Okay. I'm trying to move you guys because I can't see. And then I'm going to try to give you an uninoculated tube versus an inoculated tube. So if you see red, okay, and you see this one, then you have to say, okay, I can say this one has bacterial growth, but it doesn't change like this. Okay, so that means this is a negative result. There is no change in color, no change in pH. The bacteria cannot ferment the sugar, whatever sugar it is. So I'm trying to give you, I'm going to go, that's why I go in early. If she did not set out a tube like this, then I'll try to add a tube in so that you guys can compare the color and to see if there's bacterial growth or not. If I don't add this and I give you a tube similar to this over here, then you could have said, Professor, there's no bacterial growth. So I can't tell you what the result is. And that's perfectly a good answer. Okay, but I'm trying to avoid that, so I'm going to give you an uninoculated tube, and then you can compare. Question? Oh, this is the one, huh, Raven?
Yeah.
Okay. So tube A and B, what test is that?
Isn't this the biotolerant test?
Yeah, bioesculent hydrolysis test.
The hydrolysis test.
So there are 2 parts to it. So you have to give me both, okay?
Okay.
It's the bioesculent hydrolysis test. What are they selecting for?
To see if the enzyme is there to the enzyme esculent or like hydrolyze the esculent and turn the agar black.
I don't think there's an enzyme there. You can double check me.
Let me check.
Because I don't recall having an enzyme name. So that means this is not an enzymatic reaction. Double check me.
Mm-hmm.
But what this test looking for is to see if the bacteria are able to tolerate biosol.
Oh, okay.
And I think typically gram-negative bacteria tolerate biosol.
Correct.
So for this one, you check for growth first. If you don't see growth, then you can, and the loop was perfectly cool, everything was correctly inoculated, and then you can say the organism cannot tolerate biosalt. That's why they can't grow.
Yeah, you're right. There's no enzyme.
There is an enzyme?
No, there isn't.
Yeah, there isn't an enzyme, yeah. So it's not an enzymatic reaction.
Okay.
So you see grow in both tubes, so you can say, for one point, you say, well, there's growth, so the bacteria tolerate biosol. And then for the second point, you say, tube A, they couldn't hydrolyze, or they use water to break bond. They couldn't hydrolyze the substrate esculent. However, because there's no color change. However, in tube B, they're able to hydrolyze esculent into esculetin. Esculetin react with ferric citrate, an indicator, and give me this dark precipitate. And then on the plate, okay, you guys know this. This is a catalase test. Okay, the name of the enzyme is catalase. And then complete these reactions. I add hydrogen peroxide. If the organism have catalase, then it breaks down to blank plus water.
Oxygen.
Oxygens. And then the last one, interpret results. Okay. Site A has bubble. Bubble is oxygen. So organism in site A have the enzyme catalase, able to break down hydrogen peroxide. Side B, there's no bubbles, so this reaction does not happen. So this organism do not have the enzyme catalase. I could ask you, why do they need catalase? What's the purpose of having the enzyme catalase? Okay, and you say hydrogen peroxide is toxic to them, so they need catalase to break it down to non-toxic form, oxygen and hydrogen peroxide, in order to survive. Okay, so on this side, on side B, when I add hydrogen peroxide, those bacteria underneath is probably being killed right now. Right, 19 is on the motility, right? So I asked name two other methods to determine motility.
Wet mount and
Wet
a
mount?
hanging drop. Yeah, you could use a wet mount and a hanging drop slide to see how
Exactly.
bacteria can grow
Mm-hmm.
and move.
Hey, Aaron, can you help? I'm thirsty. Can you help me interpret result for the motility test, please?
You want me to?
Yes, please.
Oh, I was like, wait, what? So let's see. So on interpret results, so A and B, so the motility test, you can see on tube B, when it was inoculated, it's just on one streak. On A, you see it going everywhere. So it's starting to move.
So in terms of positive and negative tests, which one's positive?
Positive, tube A is positive for motility.
Excellent. Very good. Now, on the plate, okay, they added, name this test. Anybody know the name of this test?
This is a Sarch hydrolysis.
Very good. why do you say that it's a starch hydrolysis? What is it about this plate that help you come to that conclusions?
Because this is the one where you use the iodine to see if it creates a halo around it.
Exactly. Okay. So what is the name that break down starch?
Amylase or something like that.
Amylase.
Amylase.
If this is A on the left and B on the right, which of these organisms has the enzyme amylase?
B, because they formed a halo around the bacteria.
Exactly. Just like that. Okay. So on Monday, Raymond, after we go through result and I have the station set up, go around your salvo with your group. Make sure you put a 4-minute and just run through each of the stations.
Okay, so on Monday you're gonna put exactly these ones in person?
Yes.
Okay, makes
Not
sense.
exact, some kind of form.
Yeah, okay. But we could work on
Something
we could
similar.
work on it in groups and like time each other?
Yeah.
Okay, perfect.
But let everyone talk, you know what I mean? Like
Yeah.
take turn.
Mm-hmm.
Because they have to also be able to express or put things into words.
Correct.
You know, and then you become the checker. You check them.
Correct.
You know?
Mm-hmm.
So that's the best result.
Question, Professor, when when it gets closer, we're a few days away from Wednesday. This is not exactly what you show us in the mock practicum. Is not exactly what is going to be on the actual exam, correct?
Well, the question that's all you've been noticed the pattern?
Yes.
Yes. What is the name of this test? What is the indicator? What interpret result? There's not much else I can.
Okay, kind of like signs, symptoms, and diagnostics.
Yeah,
Okay.
Exactly. You can take all of these questions and make it, and then do it for every single test.
Okay.
If there's not much I can really go.
I'll get into detailing.
Yeah.
Okay.
Yeah, it's the same question asked again and again and again on different tests.
Okay.
Right. And is it going to be exact? I don't know. For example, a perfect example is this test, the KIA test, right? One day I see tube B all yellow, and I say, yes, I'm going to use that tube. That's what I want to ask. And then the day of the practicum, guess what? Tube B looked like tube C. The bacteria changed. And so I have to kind of go with the flow. I have to go in and say, oh, well, the bacteria changed. I don't have anything to replace it. I'm just going to go with it. So in micro, we have to stay really flexible. We just like go with what bacteria did. The bacteria did something. I'm just going to go with it. Yeah, so is it going to be exact? I can't tell you. It's based on what I got. When you got to donate your tube. And not just your tube, other instructor also been collecting. And so the full-timer who come in, she gonna look at all of our tube, and she'll be the one picking which tube to use on the practicum. So I don't know either, okay? I coming in a little bit blind as well until that morning.
Okay.
Yeah.
Thank you, Professor.
Yep. So this is one we're going to interpret on Monday. This is called the Klinger iron agar test. It's the KIA test. Okay. Again, I'm going to give you an uninoculated tube so that you can compare, because sometimes the red color change a little bit. The red color change because of the bacteria. The bacteria grow, cause the red to change a little bit. Okay. And so in tube A, okay, it's going to be, and for this one, you need to give me 4 results per tube. Okay, so you have to okay, how many do I need to give? This one can be 4. Okay, so the slant is going to give you the result for lactose fermentations. This is called the butt. The butt of the tube will give you glucose fermentation result. in fermentation, you're going to have an acid, a base, or a gas. So anywhere you see bubbles or cracks, so look carefully, any bubble or lifting or crack, that is the presence of a gas. So you have to tell me that as well. Okay, so 1, 2, 3. And then the fourth one is it dark or not? That is the production of hydrogen sulfide, H2S. So 4 result per tube. You got to run through it. Okay, so tube A, you can sum it up. I'm okay if you sum it up. You're going to say, Professor, you don't have to say Professor. You say tube A, going to have negative result for all because the SLAN stay red, the butt stay red, there is no bubble or crack, and there is no dark precipitate. Okay? Red SLAN mean no fermentation, no bubble mean no gas production, no dark precipitate mean there is no production of hydrogen sulfide. Simplify that. But hit all 4 for me. Okay? Versus tube B is going to be positive for lactofermentation and glucofermentation because both the SLAN and the butt has color change. There's a pH change. They change to yellow. There is no dark precipitate, so negative for H2S productions, but I see a bubble, so there is positive for gas productions. Tube C, red SLAN mean there's no lactofermentation. Yellow butt mean there's a pH change, glucose fermentation. The arrow look pointing at a bubble, and so there's gas production. However, negative for H2S production because it's not dark. There's no dark area. Tube D, SLAN is red, mean negative for lactose fermentations. It look like yellow here, okay? And hard to see down here, but it look like yellow here. So that means positive for glucofermentations. Dark precipitate H2S production. I did not point anything. You look closely at the tube. There is no bubble. There is no lifting. There's no crack. Negative for gas production. Now, if you don't see any yellow, okay, it is red SLAN, then just dark butt. You can say, I cannot determine glucose fermentation because everything is dark. And it's fair to say that. Okay? It's fair to say I can't see. Okay? There is no yellow. There's no evidence of yellow in the butt. But I don't know if there's fermentation or not. You can say cannot determine. And that's an answer. However, if you cannot see yellow, but there is a bubble there where it is dark, there is a huge bubble, okay? You have to know that there is fermentation because fermentation produces a gas, even though I don't see it. Where's the gas coming from? Look, the SLAN is red, so it can't be coming from lactose fermentation, even though I don't see yellow in the butt, but I see a huge bubble. It gotta be coming from glucose fermentations. You see, that's a little bit critical thinking there. And that separate an A from a C. The person who get an A able to tell me that, then I'm like, oh my God, that person know what she's talking about or he's talking about. That's what separate you guys out. Okay? And this next one, what test is this?
The oxidative fermentation test?
Yes, the oxidative fermentation test. A1 and A2 is organism A, B1 and B2 are organism B. So then I ask you, why do we inoculate one organism into 2 tubes?
To use one for fermentation and one for oxidation?
Yeah, you're testing for oxidation and fermentations.
So A1 would be positive for oxidation but negative for fermentation, and then B is negative for oxidation and positive for fermentation.
Correct. Make sure you mention the oil layer on top.
Okay.
Do you want us
one
to
is
mention
oxidation.
also because one stood green and one stood yellow as well, and that's why it's negative or positive?
Repeat that one more time, Lee.
Would you like us to also input that it is negative or positive for oxidation because it stayed green?
Correct.
Or do
It
you not?
is it is a pH change,
Okay.
right? Hey Raymond, what's the indicator for this? I forgot. Oh, isn't it bromthiamyl blue? Yeah, it's bromthiamyl blue.
Do you want us to know that as well, the indicator?
Yeah, because it's related to the color change.
Yeah, because that's why the broth initially starts off as green, right? Or the
Yeah. And then when fermentation happen or oxidation happen, they do produce an acid, and that's why your indicator change color.
But I remember you said sometimes it could be like half yellow and half green, right?
Yep.
And that just means
Uh-huh.
so this will be like a weak, kind of not weak, but kind of more oxidative fermentation.
Yeah.
But just say if it was like half green for the fermentation, it would be like a weak fermentation.
Yes.
Okay.
Exactly. Okay.